Go.
Hello everyone, my name is Yelisa Kodorobe and this is the Bio 225 Exercise 2 for aseptic technique and culturing microbes. I finally have a little bit of time to carry this experiment out. The sound in the background is my infant screaming, so we'll just power through and see what happens. Alright, so Exercise 2 is isolating individual colonies. As you can see, I've already taken the liberty of preparing my work area. You don't have to get too close, otherwise the glare of the light's going to bounce off the marble counter. Since there is around like 30 steps, I can't possibly remember all 30 steps, so I'm going to do my best to just read them off. The first cluster of steps, such as preparing, pouring my agar plates, already done. Gathering the developed cultures of E. coli, S. cerevisiae, and S. epidermidis. 3 plastic inoculation loops in labeled alcohol. Disposable cup we already have right here. What else? I've already washed my hands thoroughly with soap and water. I've already donned my gloves, goggles, apron, and face mask. I've already wiped my work surface area with a 10% bleach solution. I've already labeled my agar plates, which I might say are the best plates ever. These things are no lumps, no bubbles. We're great. What else? We'll start off at step number 8, which was put the corresponding cultured nutrient broths near each labeled agar plate. As you can see, this is the S. cerevisiae, better known as the yeast culture, next to the S. cerevisiae nutrient agar. The E. coli next to the E. coli, as well as the S. epidermidis next to the S. epidermidis. The next step, step 9, is to light the candle and place it on the work surface. It's already on the work surface, so we're not going to place it on the work surface. We're going to light it. Alright, it's lit. Next, we're going to remove one inoculation loop from the alcohol and shake briskly to dry. This is an inoculation loop off to the... It's right at the top left.
Yes.
It's right there.
Yes, we can see.
And I already have my bleach solution off to the side for disposal. Shake briskly. Alright, it's shook. Shake briskly to dry, so which means it's probably dry now. Next, we're going to remove, while holding the sterile loop, we're going to remove the cap. Would you mind panning back so that way? Like, I don't want to move the thing. I would rather you move the camera towards the thing rather than me move the thing so that way
Yeah,
I don't
you
move
can
the
see
camera.
the whole thing.
I know, but like I was over here.
Go.
Okay. Alright, so I've removed the cap from the... We'll do the S. cerevisiae first. Alright, so next I've removed the cap. Now I have to pass the lid through the lip of the container through the flame to sterilize. Okay, it is sterilized. Next, we're going to insert the sterile loop inside the broth, being careful not to touch the rim of the loop with my gloves, which I have been careful to do. I'm going to submerge the loop into the broth. Okay, it's in the broth. Immediately transfer the broth adhering to the loop into one quadrant of the agar plate using a zigzag motion. So we're going to do that right now. Oh, excuse me, if you can just back up a second. Okay. Alright. And we're going to just do one quadrant as directed. Okay, alright. Once that's done, we're going to place the lid on the agar plate. Put the inoculation loop into the cup of alcohol, which we're going to do right now. So that's done. We're going to flame the tip of the broth tube and replace the cap on the tube. So we've got to sterilize the lid again. Alright. Okay, next we're going to cap it since we sterilized it. And we're going to replace that back off to the side where it's nice and safe. Next we're going to remove the inoculation loop from the alcohol and shake to dry. So we're going to shake vigorously to dry. That's done. We're going to open the lid of the agar plate again and touch the loop to the edge of the inoculated quadrant to transfer microbes to the adjacent quadrant on the plate. So we're gonna, the quadrant that we just inoculated, we're going to kind of do the zigzag pattern again into a different quadrant, like so. And once that's done, we're gonna return the lid to the agar plate. So every time we do this process, we do that. Next, we put the inoculation loop into the cup of alcohol. After 20 seconds, remove the inoculation loop. Since I've got three inoculation loops here, I'm just going to rotate them, because if I'm waiting 20 seconds in between every inoculation, it's going to take forever. Alright, so I'll leave that one in the alcohol for another 20 seconds, and shake vigorously to dry. Next, I'm going to do the same thing, this time for a third quadrant. So we're going to do that. Hopefully that's done. I'm going to replace the lid. And we do that one last time, but this time the quadrant will not, in fact, we're not going to, the third quadrant that we did is not going to make contact with the last quadrant. All right, that's all there is. I'm gonna do the other two cultures off camera because of time constraints. This is basically what I demonstrated was what I hope is the proper technique. I tried to follow the directions. Thank you.